So we're moving on to talk about the digestive system, and you'll get a lot on the anatomy of the digestive system during lab. I will cover a little bit of anatomy, but since you're going to get a deeper look at it during lab, I won't cover it too much. We'll just cover the anatomy that we need to make sure that you're on track with as it relates to certain physiological things that we're going to be discussing that happen in the body. So really quickly, just to remind you, most people know this already, but the duties of the digestive system include to take food in from your environment, that's called ingestion, to then break that food down. That food consists of lots of organic macromolecules, and those molecules are way too big to enter into our blood directly. So we have to go through a process of what's called digestion, where we break down those macromolecules into individual, for example, amino acids or individual sugar molecules or individual nucleic acids. And then they're small enough that we can do step 3 of the process, which is to absorb those nutrients, those organic molecules, and get them delivered into the blood. As always, there's going to be some portions of the food we eat that are not used, that we do not have a need for. Those we eliminate in the form of feces after a couple days. Now, the different types of duties that are found, or the different types of mechanisms that are found to accomplish these duties include a bunch of different things that we're going to be talking about, including a process called motility. That has to do with coordinated contractions of smooth muscle tissue that lines the digestive tract. It has to do with secretion of lots of different digestive enzymes that help us to get the food, what we consume, broken into smaller parts. It includes membrane transport, getting items from the digestive tract, in other words, across cells and into the blood. And we're going to see that when we look at digestion, for example, there are 2 sub-processes. There's chemical digestion, where we use those enzymes to break down particles of food. And then there's also mechanical digestion, where we're just physically breaking large chunks of food into smaller chunks. But since we're not breaking apart any chemical bonds, it's not considered chemical digestion. So down here is a nice little summary. It kind of shows you the lay of the land of what the goal is here in terms of the breaking down of food. We take carbohydrates, usually long polysaccharides, we break them into monosaccharides or individual sugar groups. We take lipids that can be rather large, such as triglycerides or, triglycerides is a good example, cholesterol and things of that nature. And particularly with triglycerides, we have the ability to break those apart into some individual fatty acids and monoglycerides. We can take proteins and, using a process of chemical and mechanical digestion, eventually break that apart into the individual amino acids. And once we get down to this level, that's the point at which the molecules are small enough to be absorbed through the intestinal wall and into the blood. There's lots of different organs involved in this process. Obviously, there are a number of organs that are all connected. That's how we pass the food from point A to point B to point C to point D. And those include the mouth, the esophagus, the stomach, the small intestine here, the large intestine. Those are all regions of what's called the digestive tract. It's considered a tract because these are all continuous features. All of these organs are connected with one another in one long tube. By comparison, we also have some other organs that are not part of that tube, but they oftentimes will dump substances into that digestive tract tube. These are called the accessory organs, and they include the pancreas, the liver, the gallbladder, and things like the salivary glands are considered accessory organs. That's a good enough list. So the main part of the digestive tract is where the food actually moves through the tubular feature. Accessory organs are there to assist in the digestion usually by providing some sort of chemical substance such as saliva, or if you're the liver, maybe it's providing bile, or the pancreas, which provides lots of digestive enzymes. There are multiple stages to the digestion process. The first stage of the digestive process is simply to get a large hunk of food broken apart into smaller chunks. So it's getting this apple that the guy is eating here into small enough chunks that they can pass down through the esophagus and enter into the stomach. We also want to reduce the size of the food pieces through this mechanical process, because the more I reduce the size of the individual food particles, the more I increase surface area. The more surface area I have, the more rapidly I can progress through chemical digestion. So in other words, we're taking one large thing one large blob that may only have surface area along the outside, and if we divide it into 9 smaller blobs, I've increased the surface area to volume ratio. That means there's more locations where enzymes could act upon this food and during chemical digestion break it apart, chemically speaking. Mechanical digestion includes things like manipulating your food in your mouth, okay, letting your teeth chew your food into smaller bites. It also includes your stomach. Your stomach does a lot of churning, and that helps to break down food into even smaller bits and pieces. And the small intestine also does some kind of a sort of churning sensation that helps to break things down mechanically. So again, the idea with mechanical is we're not breaking any chemical bonds. We're just taking big chunks of food and making them smaller and smaller to increase surface area. Once I've got the surface area sufficiently high, then I can start to work on the second stage of digestion, which is the chemical stage of digestion. And this is the process where we have all those different hydrolysis reactions that split organic molecules apart. Remember, hydrolysis literally means to split apart using water. Lysis is to split. Hydro relates to water. So we're literally using water to split apart organic molecules. So if I had 2 amino acids strung together, hydrolysis would be the chemical mechanism I would use to break that bond, and water is consumed during that process. Now, we're going to be seeing that there are lots of different types of reactions for hydrolysis reactions for breaking down sugars, for breaking down nucleotides and polypeptide chains, and for breaking down fatty acids. And these substances are manufactured in a number of different places. The salivary glands make some of the chemicals responsible for hydrolysis. The stomach makes one. The pancreas makes a whole bunch. And the small intestine has a number of different enzymes that help to break down food. One important thing I want to highlight here, and I will come back to this later when we get to the liver and talk about it even more, is the fact that the bile which the liver produces is not involved in chemical digestion. It's actually involved in mechanical digestion via a process we call emulsification. I will reserve talking about that until later, but we'll talk about it in more detail. So the final stage of digestion is once I've chemically broken apart all of these organic molecules, I've gotten them as small as they're going to get. Now I have these small molecules that I can absorb. Okay, so these small molecules can be absorbed through the wall of the small intestine and then introduced into the blood. Now I should point out that there are some molecules that when you eat them in the first place are already relatively small. And those that are already relatively small when you eat them in the first place don't even need to go through this chemical digestion process. So things like certain free amino acids that are found in your foods, minerals, such as magnesium, for example, cholesterol molecules are small enough to be absorbed without digestion. Water is, vitamins, etc. So the first place that we have digestion occurring is in the mouth. And that should be no surprise, really. The mouth is also called the buccal or oral cavity, and it consists of your teeth, the tongue, which helps you to taste the food to make sure you're not eating anything poisonous, but it also helps to move your food around in your mouth so you can position it between your teeth and chew it or masticate it is another word for chewing. You can masticate it into smaller pieces. Again, we're trying to increase that overall surface area. We have a palate at the top of the mouth, up in here. And that palate allows us to have our mouth be a separate cavity from our sinuses, and that allows you to chew your food and still breathe at the same time, so you don't have to chew with your mouth open. You have a lot of different teeth, different types of teeth that you'll learn about in lab: incisors, canines, prenolars, molars. You have 20 what are called baby teeth, or what we call deciduous teeth, that you lose as a child. And those are eventually replaced with 32 permanent teeth. And I'm not going to go over the different types of teeth. You'll get to do that in lab. I've already mentioned that mastication is the process of chewing. Okay, now in the mouth you have a number of salivary glands. And the salivary glands are situated in a number of different places. I'm going to focus here on the salivary glands that are immediately inferior to the tongue, but don't forget there's also ones in your cheek and lower down under the tongue. Again, you'll learn about those in lab. The main characteristic that saliva does that contributes to chemical digestion is that the salivary glands manufacture an enzyme called salivary amylase. Salivary amylase is an enzyme that has the ability to break down starch, and it works very well in a neutral pH, which is what your mouth is. Your mouth has a relative pH of about 7, give or take, pretty neutral pH. However, salivary amylase does not work well at other pHs. So when you chew food and it comes into contact with saliva, if there's starches in there, some of those starches will begin to undergo hydrolysis because of the activity of the salivary amylase. As soon as you swallow that food, it goes to your stomach. Your stomach has a much more acidic pH. At the point that that salivary amylase comes in contact with the stomach contents, it becomes deactivated. It stops working because the pH range is wrong for that enzyme to be active. Now, the salivary glands also make another enzyme, and that enzyme is called lingual lipase. Some books call it salivary lipase. Same thing. Lingual lipase, or salivary lipase, is an enzyme that has the ability to digest fats. So triglycerides, for example. It's interesting in that it is not active at a neutral pH. It only becomes active at an acidic pH. So whereas salivary amylase was active in the mouth, but it stops working when it reaches the stomach, lingual lipase does the reverse. Lingual lipase may come in contact with fats in your mouth, but it will not hydrolyze any of those fats until you swallow that food and it comes in contact with the acidic stomach. And that acidic environment in the stomach then activates lingual lipase, and it begins to digest roughly about 10% of all the fats that you consume. Now, there are other components to the saliva besides these two enzymes. You know, saliva has a good quantity of water in it. It has mucus in it, and that mucus helps to kind of stick together all the food that you've chewed, and it forms it into sort of a ball-like structure called a bolus. And that ball-like structure is usually kind of oblong shape. And because it's stuck together with mucus and kind of lubricated and covered by that mucus, it makes it easier to pass that bolus down your esophagus. It makes for easier movement. Saliva also has lots of immunoglobulin A antibodies present, and these, as we learned before, are present in mucus, and they're very good at attacking or tagging bacteria for destruction by things like macrophages. So serving a surveilling role here to make sure you're not consuming bacteria and to neutralize whatever bacteria you do consume. There's also lysozyme, which you learned about before, is a special enzyme that breaks down the peptidoglycan cell wall of bacteria. That too is found in the saliva. And all of these combined together perform a number of different roles. And again, they do it all roughly at about a neutral pH of 7, maybe 6.8 at the lower end. I want to talk really quickly about salivation. How do we get saliva production going in the mouth? And that's a process that happens, it can happen a number of different ways. We all know that saliva production increases when we're actually eating. And that's because the touch, the taste of food, the touch of it in your mouth, the pressure receptors in your mouth, those are all stimuli that sends off signals to the brain and induces salivating activity. And it really, if you want to get really specific, the signal is sent to places like the pons and the medulla oblongata. You have what are called salivatory nuclei in those locations, whose principal job it is simply to stimulate the production of saliva in the mouth. And both the parasympathetic and the sympathetic nervous system come into play here. The parasympathetic nervous system is responsible for producing the watery part of the saliva, if you will. Some books call it the thin part of the saliva. It's the watery part of the saliva. The sympathetic nervous system, your fight or flight response, tends to stimulate the production of things like mucus. So it's kind of like the thick part of the saliva. This is why, by the way, if you ever have a fight or flight response, if you've noticed that your mouth got really dry and sticky, it's because in a fight or flight response, your parasympathetic nervous system is being suppressed. And so you aren't getting any water production for your mucus. You're only getting for your saliva, you're only getting the mucus part of the saliva being produced. And so it's a very thick saliva, makes your mouth feel very dry. Now, we also know that higher brain centers can influence when we salivate. For example, just thinking about a food, even when the food isn't present, can oftentimes generate salivation. The last series of slides here I'm going to talk about before we launch into the stomach is to just give you a general anatomical overview of what the digestive tract consists of. There are multiple layers to the digestive tract. We're going to go from the inside, from here. I'm going to work my way outward this way and tell you the different layers. On the innermost part of the digestive tract is a mucosal layer. And that mucosal layer has several distinct layers of tissue. It includes a stratified squamous epithelium. Should make sense. We get lots of abrasion as food passes by through the digestive tract, so we want to have lots of layers of cells to protect. And remember, what I'm talking about here when I say the digestive tract, I'm talking about this is basically the generalized lay of the land for the esophagus, for the stomach, for the small intestine, for the large intestine. They all have these components. Sometimes one component may be missing or a new component may be added, but for the most part, they all follow this structure that I'm outlining for you here. So deep to the epithelial layer of the mucosa is a thin layer called the lamina propria. This is made up of areolar connective tissue, and it's there to nourish the epithelium above it. And then there's a thin layer of smooth muscle tissue called the muscularis mucosae. So just a thin little layer of smooth muscle tissue. It's a little hard for me to point out these layers to you in this picture just because of size, but so you can see here, the lamina propria is this kind of greenish area here. This would be where I'm putting the stars, is the epithelium of the mucosa, and then right in here would be where your thin layer of smooth muscle tissue is, the muscularis mucosa. Going out farther, so we're getting farther away from the lumen and out toward the edge, the next thing we encounter is submucosa. And the submucosa is also a layer of connective tissue. It's loose connective tissue. It helps to assist in lubrication of the lumen via the production of mucus, and we'll see more about that later. The outermost of the three layers of the digestive tract is the muscularis externa. And the muscularis externa is the layer that has a large amount of smooth muscle tissue in places such as the stomach, the large intestine and the small intestine. Around the mouth area, this muscularis externa layer is made of skeletal muscle tissue, so that it's under conscious control, and then it's smooth muscle tissue as you get deeper into the digestive tract. That muscularis externa has two layers to it. It has an inner layer that runs in circles, okay, and then it has an outer longitudinal layer. So here's my, do they have it here? Yeah, you can see it down here. Here's the inner circular layer of smooth muscle, and then the outer layer is running this way. It's running longitudinally, up and down. Later on, we'll see that the stomach has a third muscularis externa layer, and we'll learn why that is when we get to the stomach. Lastly, we got to wrap the outside of the digestive tract with just a thin layer of little connective tissue just to kind of hold everything in place. If that connective tissue thin layer is found above the diaphragm, we call it the adventitia. And again, this is just a thin layer of connective tissue that kind of protects the digestive tract, holds it in place. Below the level of the diaphragm, we call that same layer of connective tissue the serosa. Okay, and it has a slightly different composition of cells in the serosa than the adventitia. And we're not going to worry about the specifics of that. Just know it's adventitia above the diaphragm, serosa below. And remember again that all parts of the digestive tract follow this basic scheme of having your mucosa, the submucosa, the muscularis layer, and then this either adventitia or serosa, with minor modifications now and then. But for the most part, it's these three layers plus a little connective tissue on the outside. There's also interspersed among all these layers a very, very important collection or network of neurons called the enteric nervous system. The enteric nervous system helps to orchestrate motility. In other words, when do the smooth muscle cells in my digestive tract contract and when do they relax? It also helps to orchestrate secretion of enzymes. And it helps to orchestrate perfusion, or when blood flow is increased or decreased to the digestive tract. The enteric nervous system is estimated to hold about 100 billion with a B neurons. That's about the same amount as we estimate are found in your spinal cord. So you have as many neurons present in your digestive tract as you do in your spinal cord. They are divided into two plexi, or two networks, if you will. The submucosal plexus sits immediately outside of, or below, if you will, the submucosal layer. And the myenteric plexus sits in between that outer longitudinal and inner circular layer of smooth muscle tissue. As you might guess, the myenteric plexus is the one that sort of orchestrates motility activities. The submucosal plexus tends to be more interested in orchestration of perfusion and secretion activities. Now, we can control behaviors of the enteric nervous system and the reflexes that it encourages via what are called both short reflexes and long reflexes. Short reflexes involve the stimulation of, say, secretion of enzymes just because of the actual presence of food in the digestive tract. Later on, we're going to see that, for example, when a food bolus enters into your esophagus, the simple stretching of the esophagus because of the presence of that food actually induces smooth muscle contraction all on its own. That's an example of a short reflex. There are also long reflexes, and this is when the enteric nervous system communicates with the central nervous system via the autonomic nervous system. So this is when you have communication back and forth between the digestive tract and the brain to coordinate activities. But many of these activities happen by short reflexes. You don't have to have the brain directly involved. The enteric nervous system does plenty of orchestration all on its own. Okay, that's good enough for now. The next thing we're going to do is we're going to look at what happens when I swallow food and I start getting into the true tubular forming part of the digestive tract.